Thank you so much for the opportunity to present this work. My name is Emmanuel San, and I'm a bioinformatician here at the DHVI in the WIHI lab. Our lab specializes in computational analysis of B cell development and immune responses to HIV and other pathogens. We also develop analytical tools and methods. We have developed and published a tool called Armadillo that is free and publicly available online for computation of probability of mutations in antibody sequences. And yeah, with that, let's get right into it. I'll give a quick overview. Previous speakers have introduced this quite a bit. So antibodies originate through a process known as VDJ recombination, where selected V, D, and J sequences from the immune repertoire recombine to form heavy chain genes, while V and J genes recombine to form light chain genes. These chains then assemble and create a functional antibody. Upon exposure to antigen, these antibodies undergo affinity maturation, a refinement process that enhances their binding affinity. In acute infections, the maturation occurs relatively rapidly. However, in chronic infections, the process is significantly slower, such that the evolutionary dynamics can be observed and studied. Although rare, in chronic untreated infections, a unique subset of antibodies known as broadly neutralizing antibodies, or bnAbs, can emerge. bnAbs can neutralize diverse HIV strains. And a good example is the CH235 and N6 that both can neutralize over 90% of the currently characterized strains. And the figure on the right shows you the HIV envelope and the antibodies here. The longer arm is the heavy chain and the shorter arm is the light chain. So to ground us a little more, this is the CH235 antibody. The CH235 is a bnAb targeting the CD4 binding site of HIV. It was first detected 14 weeks post-infection in the patient and underwent a 6-year maturation process, ultimately reaching maximum potency and breadth at week 323. To characterize this lineage, sequences from 17 time points were analyzed. The tips of the tree that you see show the sequences that belong to this lineage, while the nodes labeled in blue show you the intermediate sequences, which represent the key time points in the evolution of this lineage. The thick black line traces the antibody's developmental trajectory from the UCA to CH235, which is the fully matured and potent form of this bnAb. Mutations acquired at each stage are listed on the side, with H indicating the heavy chain mutations and L indicating the light chain mutations. The color scale represents the probability of acquiring any of those mutations, while the number in the circles represents the total number of mutations acquired at each of these stages. So following the detection of bnAbs, HIV vaccine research has focused on training the immune system to induce and mature bnAbs in humans and in non-human primates. The mutation-guided vaccine design strategy aims to design antigens that enhance the immune response by selecting antibodies with targeted mutations at each stage of affinity maturation. To achieve this, bnAb clones are identified and analyzed to reconstruct their evolutionary history, tracking the sequence of mutations acquired during affinity maturation. This information is then used to design priming and boosting immunogens, which can bind the antibody at each developmental stage. The unmutated common ancestor represents the naive or secreted form of the antibody. It is therefore used to design the priming immunogen, while the intermediate sequences are used to design the sequential boosting immunogens that bind the antibody with the desired mutations. This approach enables the body to induce the desired antibodies, proliferate them, and shepherd them to maturity, thus enhancing the adaptive immunity. The evolutionary pathway of antibodies can be traced through phylogenetic analysis, providing insights into their development and potential for vaccine design. So the objective of phylogenetic analysis and bnAb studies is to determine the most probable precursor, to infer the intermediate sequences, and to determine the mutation profiles of the UCA, intermediates, and clonal members. To achieve this, our lab has predominantly used clonalist, an integrated suite of statistical methods for analysis of antigen receptor rearrangements and somatic genetics. Clonalist will annotate and partition your sequences. It will infer the UCA for you and the intermediate sequences, and then calculate the phylogenetic tree using both the heavy and the light chain sequences. The annotation and partition can be performed from the command line, while the UCA and tree inference are performed using clonalist's GUI. However, recent findings reveal an underrepresentation of IG genes in gene databases, like one of the speakers mentioned earlier. And this is particularly true for rhesus macaques, a non-human primate that has long been the focus of research to understand the immune responses to human pathogens due to their close genetic relationship to humans. Their genome shares 93% similarity with the human genome and 97% similarity in the coding regions. Recently, updated gene databases such as ASC Germline database and KimDB have been published, and most sequences not present in these databases continue to be identified, making this an ongoing area of research. And new improved IG-specific mutation models like IG5ML's HP19 model that incorporate AID targeting during tree inference have also been developed. Unfortunately, clonalist IG gene databases and tree inference models cannot be developed to updated to utilize these resources due to compiled and closed source code. So to address these challenges, we have had to modify our analysis workflow. We have integrated new tools, including IG Discover to infer new alleles in our dataset, parties to perform annotation and partitioning using these new databases, such as KimDB and ASC, Lineages from Eric Mattison's lab at the Fred Hatch to infer UCAs. However, we still needed a way to perform paired chain phylogenetic tree inference. And that's where Dowser comes in. So while both Dowser and clonalist are able to perform paired chain tree inference and generate for us the intermediates that we need, they too are slightly different in the sense that clonalist, although being able to also infer a full-length UCA and provide a GUI, which means you can use it without any programming experience, is not actively maintained anymore. The gene databases are not updated. The UCA and tree inference are limited to the Windows GUI. You can only analyze 99 sequences at a time, and it offers no provision to for you to use your own UCA. And that on the flip side, Dowser allows us to script and run it from the command line, making it suitable for workflow integration. It is actively maintained, has great user support, and is open source, with its only limitation being the inability to infer a full-length UCA that includes the N-nucleotide regions. Although we know that the developers are actively working on this, and we are hopeful that this feature will also be available soon. However, the cool thing about Dowser was that while it did not infer the UCA for us, it allowed us to provide our own. And so for the rest of this talk, I'd like to focus on our evaluation of Dowser and how we are currently using it in our BCR phylogenetics and intermediate sequence inference. So to evaluate Dowser, we used two previously identified and well-characterized bnAb clones, DH270 and DH570. The original trees and intermediate sequences were inferred using clonalist. We compared the tree topologies using the Robinson folds distances and the clades that were identified. The Robinson's fold to RF distance counts for you the number of splits between two trees. And we also compared the intermediate sequences generated using the two methods with a special emphasis on key mutations. So first, we assessed Dowser's ability to reconstruct the DH270 lineage. And the DH270 is a V3 glycan bnAb with the DH270.6 as the most potent member of this lineage. The samples from 7 time points spanning a 5-year period were analyzed. The bnAb developed full breadth and potence after 3.5 years. When we compared the clonalist tree to the one produced by Dowser, we observed that the topologies were identical and with an RF distance of 0, confirming that indeed the two trees shared the same branching patterns. So next, we compared the intermediates generated by clonalist and Dowser. Although over 42 mutations had emerged along the maturation pathway of DH270, Swanson and colleagues have identified the 12 most important mutations that are critical for breadth and affinity of this bnAb, which they dubbed the minimized DH270. 2 of these mutations, shown in brown, are extremely improbable, which means that they're very difficult to induce, and another 6, shown in red, are also improbable, but with a slightly higher probability. So we checked for the presence of these mutations in the intermediate sequences. To our delight, Dowser was able to recover all the critical mutations that clonalists had earlier detected and agreed with clonalists on the location along the branches of where those important mutations occur. One interesting observation, though, was how Dowser treated the S27Y light chain mutation. The S27Y is a highly improbable mutation, and this is likely due to its need for 2 base changes, AGT to TAT, to achieve this mutation. Dowser highlights an intermediate step S27N, which is a change from AGT to AAT that finally transitions to the S27Y in the next step. Remarkably, this is in line with other research that have shown that the asparagine, or N, and cysteine, C, are possible pathways to the tyrosine, Y. This kind of information is therefore valuable for immunogen design, where an immunogen can be designed to select the S27N as a precursor for the S27Y. Next, we wanted to see how Dowser could perform as the number of sequences increased, whether we could use it to infer an accurate phylogeny for rhesus macaques, as highlighted earlier. We therefore tested Dowser on the DH570, a rhesus macaque antibody lineage, which originally comprised 32 members or sequences. The 2F5-like EMPA peptide antibody was sorted by flow cytometry from PBMCs of a rhesus macaque between weeks 43 and 144 of antigen challenge. Using the old or new data from this lineage, we were able to explore the effectiveness of Dowser. We compared the DH570 tree topologies of Dowser and clonalist. The topologies were largely similar, except for a few differences in the relationships between the clade members. For example, if you look at clade 2 and 4, you will see a slight twist in how the members are aligned there. The differences, we believe, are likely due to more sophisticated mutation model used by Dowser and the updated IgGene library that we used to infer the UCA prior to our tree inference. Once we were satisfied with the results from the old sequences, we proceeded to use Dowser to infer a new tree for the DH570 lineage with newly sampled data. At week 632, 12 years later, new samples were obtained and sequenced. We combined the old and new data, an additional 45 sequences to make it 77. and inferred a new tree. From the tree, we can see that DH570.18 had mutated further. It is possible that these new clonal members that have further evolved could have gained higher affinity and breadth. This will need to be confirmed in the lab. So to conclude, the trees inferred by Dowser and clonalists were highly similar. The intermediate sequences that it generated were also comparable to clonalists and contained the key mutations that were required for potency and breadth. And as a precursor, the UCA represents the secreted form of the antibody prior to any somatic hypermutation, making it a crucial element for the tracing of a complete evolutionary pathway of the bnAb. The inference of an accurate and complete UCA with nucleotide regions therefore remains an important challenge for Dowser. Nevertheless, we confirmed that Dowser was indeed a robust and reliable tool for B-cell lineage phylogenetic reconstruction, providing a valuable asset for advancing mutation-guided vaccine design strategies. And with that, I'd like to thank the DHVI, my PI, Kevin Weehee, members of the Weehee Lab, particularly Elizabeth, Shavani, and Josh, who were key contributors to this work. I would also like to extend my appreciation to Ken and call for their support and wavering throughout the integration process. And finally, to the Immcantation community and Kleinstein Lab that have spent countless hours organizing this event. And last but not least, I'd like to also thank my wife who stayed up to attend this call. And thank you all for your attention. And with that, I'm happy to take any questions that you may have.
Thank you very much for your presentation. There is one question on the chat. Dowser support multiple tree construction methods. What method did you test here, and did you test other methods supported in Dowser?
Yeah, that's a very good question. We tested all of them. The most accurate, or the one closest to clonalist was RAXML. And so the trees that I have shown were inferred using RAXML.
Thank you for your presentation. We are going to move to our next speaker.